Maybe we can begin. My name is Chris Fanta. I'm delighted to welcome you to our asthma grand rounds and have the opportunity to wish you all a happy and fulfilling new year. And the subject of our asthma grand rounds is an area of medicine and asthma and genetics that I think 20 years ago was not known. The topic of microRNA and asthma. And to talk about this subject, of course, we turn to Dr. Kelan Tantisira, who's been a great friend of Asthma Grand Rounds and a frequent speaker. He is a geneticist, he is a pulmonologist, he is a pediatric pulmonologist. He's a member of the Channing Division of Network Medicine and the Pulmonary and Critical Care Medicine Division at Brigham and Women's Hospital. And I very much appreciate you talking to us, introducing us to the subject of microRNA and asthma. Dr. Tantisira.
Thanks, Chris. I appreciate the invitation to speak with you this new year. And indeed, I will be speaking today about microRNAs and asthma. Chris gave the topic as a primer, and I think we'll try and keep it towards that, giving more global messages as well as a few specific examples rather than trying to delve too deeply into each aspect of the literature. Now, this is broadly what I'd like to speak about in the short hour that we have today. I want to clearly discuss what microRNA is and what does it do. We'll talk a little bit about just general global asthma microRNA association studies and talk about microRNAs as key network hubs, key network modulators as part of those association studies. We'll also talk about microRNAs in the context of what do they do for a specific cellular function? How might they regulate specific cellular function in specific contexts? The area that I'm most closely involved in microRNA research is circulating microRNA, and we'll see what we can describe about circulating microRNA as disease biomarkers. And finally, a nice ending topic is microRNA as a potential for therapeutic targets. So what is microRNA? Well, globally, microRNAs are also known as microribonucleic acids, also known as miRs or miRNAs, and I typically refer to them as miRNAs. They're short RNAs. They're only 21 to 23 base pairs in length. And there's very, very strong conservation. So the microRNAs were first described in yeast, and the initial microRNAs that were described in yeast are, in fact, conserved into humans. They're epigenetic regulators of gene expression. And what do we mean by epigenetic regulators? Well, they're regulators that are not coded for by heritable changes in DNA. So they are what they are, and as I mentioned, that goes along with the strong conservation across species. Now, miR-base21 is the microRNA database that is most commonly cited, and currently it has 28,645 entries across all species. In the humans, that equates to about 1,881 precursors with 2,500 or so mature known microRNAs. The convention for naming microRNAs is pretty simple. It starts with the organism of choice, and then just a miR for microRNA, and then the order in which the microRNA was discovered. So miR10, for instance, was the 10th microRNA described in humans, and the 10A describes the fact that there is a family of similarly related microRNAs, so there's also a 10B and a 10C, for instance. So what does microRNA specifically do? Well, microRNAs are produced as pre-microRNAs in the nucleus, and then they're exported by a complex and binds to an RNA silencing complex. And if you think about silencing, that's exactly what it does. If it's perfectly complementary, in other words, all the base pairs perfectly match, it results in gene cleavage, total elimination of any gene product. If it is imperfectly complementary, so that there might be one or two base pairs that don't match, which is the case in most human studies, then it results in translational repression or decreased in gene expression. So microRNAs are one of nature's ways to modulate gene expression overall. Now, in the context of studying microRNAs, words that you might hear are microRNA mimics and antagomers or antagonists of microRNAs. And exactly what do? Well, similarly to regular microRNA, as it binds to the gene, the 3' end of a gene, microRNA mimics are designed to look like that and also bind to the 3' portion of the RNA and result in translational repression as well, whereas antagomiers specifically antagonize the microRNAs and therefore inhibit binding and allow for normal translation to occur. Now, microRNAs are generated typically within a nucleus of cells, and then they're exported in a variety of mechanisms. Certainly, a couple of those relate to cell death. In terms of apoptosis or cellular necrosis, the microRNAs are freely released, in which case then they're bound to proteins in the circulation. MicroRNAs also function by active excretion during times of cellular processes such as inflammation, in which case these things are excreted in nanovesicles. Most commonly known of those nanovesicles are exosomes, but there are other small particle vesicles containing these microRNAs as they go about. And so microRNAs typically are best known for intracellular communication, whether it's in a local milieu or a distant milieu. This is an example of microRNAs in a cell that's adjacent to another cell. And you can see that the microRNAs are extruded either in these exosomal bodies or through other processes, and they're taken up in a neighbor cell. So important in cell-to-cell regulation, and then the microRNAs tend to do this, the gene translational repression in genes. Now, you notice one thing is we said that many human microRNAs are not perfectly complementary to their targets, which allows microRNAs to bind to multiple genes and regulate multiple genes simultaneously. So let's talk a little bit about microRNAs in human asthma and the association studies that have been performed so far. Now, there have been a wide variety of microRNA association studies. This is actually not comprehensive, but this is a good representation of the studies that are out there. I think you'll notice a couple of things in general, outside of the fact that the fonts are relatively small here. One is that the sample sizes are relatively modest in terms of case control association studies. Some of these involve only 4 or 5 cases or 4 or 5 controls, and a couple of them get up into the 100 type of range, but really fairly modest overall. The second is that there are many studies that do use arrays, but some studies that only interrogate 5 or 6 microRNAs, in which case we're not able to get the full sense as to what microRNAs are truly associated. Having said that, I wanted to go over some exemplar studies. This is a study that was published about 4 years ago in the Blue Journal. This is taken from airway epithelial biopsies and brushings of 7 cases and 7 controls using an Affymetrix microRNA array. The important thing to note is that the healthy controls are shown here on the left, the asthmatics are shown on the right, and this is what we call a typical heat map plot for microRNA differential expression, where red is upregulated genes, and in this case upregulated microRNAs, and blue is downregulated microRNAs. And you can see that there is a series of microRNAs that look to be very differentially expressed between healthy controls and asthmatics, both upregulated and downregulated. The nice thing about these microRNAs is that we can infer targets for the microRNAs and therefore infer biology that is influenced by this group of microRNAs. And indeed, for this case, these microRNAs in these individuals seem to regulate the IL-1 beta pathway down through the NF-kappa B signaling. So that's just an example of a microRNA study. There are similar studies that have been like this. Again, another heat map study here showing differential expression between asthmatic cases and controls. In this case, they actually found over 200 microRNAs that were differentially expressed between healthy cases and controls, again, using airway epithelial cell brushings in these individuals. The nice thing that these individuals also did is they subsequently looked and said, well, you know, we treat these asthmatics with corticosteroids. Is there differential expression of microRNA that may result from treatment? And in fact, in therapeutics, there were not hundreds, but there were 9 strongly differentially expressed microRNAs just from administration of inhaled corticosteroids. They did not look specifically at pharmacogenomic outcomes, so this had nothing to do with treatment response, but steroids themselves can also regulate microRNAs. And we'll get into this a little bit more later on. The final case control study that I wanted to show was something I thought was very interesting. This is a principal components plot. So instead of showing individual microRNAs that are differentially expressed, this shows the sum of microRNA expression in cases versus in control. So globally, if you look at all microRNAs and just plot them out in 3-dimensional space, this is what you get. And the projection basically shows that there are clear differences globally looking at microRNAs from asthmatics versus healthy controls. And interestingly enough, there's a line of division right here down the middle across principal component number 1. And you'll notice that there are actually 2 samples that seem to side with the healthy controls rather than the asthmatics. Now, when they went back and looked at these 2 individuals, they found that these 8 individuals were all highly atopic asthmatics, and these 2 individuals were non-atopic asthmatics. And therefore, it might be that principal component number 1 determines atopy versus non-atopy in terms of asthma, and principal component number 2 actually might have something to do with atopic status. So we talked about microRNAs in the global context of human association studies. What about as key network modulators? This is a social network, but we'll talk about microRNAs in terms of gene networks in this particular case. And these individuals looked at a publicly available data set looking at gene expression data between cases and controls. And they found 274 asthma genes that were differentially expressed between cases and controls, and they mapped them to the human protein-protein interaction database. And the vast majority of genes mapped to one huge, gigantic cluster of differentially expressed genes. And you see some other genes that were mapped kind of more independently on their own. Based on using informatics databases, these investigators subsequently said, well, if we take that protein-protein interaction database and we said, well, what are the microRNAs regulating them, how does this fall out? You can see the triangles in this particular case are the microRNAs themselves. And you can see, as we mentioned before, one microRNA can regulate multiple messenger RNAs. And here you can see that this is a giant cluster of, in this case, 2 microRNAs regulating a very large cluster of messenger RNAs within that protein-protein database. There are other microRNAs here, and you can see that in some cases there's actually 2 microRNAs that regulate an individual gene. Now, because of knowing how microRNAs regulate multiple genes, you can actually look and see what are the pathway ontologic functions related to those genes. And in this particular study, they found that processes such as apoptosis and protein complex assembly and DNA metabolic processes were the highest processes differentially regulated between asthmatics and controls in terms of microRNA regulation. Now, another study that was performed, this is actually a study of allergic rhinitis and not asthma, but I liked it because what these authors did was they generated both microRNA and messenger RNA in CD4 positive cells derived from both healthy individuals as well as people with severe allergic rhinitis. And they looked at the correlations direct correlations between microRNAs as well as the messenger RNAs. This is just a differential expression, genes that were upregulated and downregulated in allergic rhinitis in CD4 positive cells versus healthy controls. And you can see that there clearly are, as expected, a wide variety of differentially expressed cells. When mapped individually to the microRNAs and saying what microRNAs directly regulate the downstream gene signaling, again, you see networks evolving where multiple microRNAs are involved. In this case, 43 microRNAs regulating 1,227 messenger RNAs that were differentially expressed between allergic rhinitis and non-allergic rhinitis in the context of CD4 positive cells. Now, the statistic that they provided was very, very interesting because each of these microRNAs, on average, regulated approximately 68 individual messenger RNA genes, again suggesting the pervasive nature of the regulation of microRNAs in this particular context. And each messenger RNA in turn was regulated by about 2.5 individual microRNAs. So as a brief interim summary, we know that microRNAs regulate and repress gene expression both at the level of individual genes as well as key network hubs. A number of asthma microRNA studies have indeed been conducted with well over 200 individual microRNAs associated with asthma to date. Most of those studies have not been independently validated, but there are certainly on the order of dozens of microRNAs that have been reported in more than one independent study. Most of the studies, as mentioned, have either limited sample sizes or limited number of microRNAs interrogated, leaving a lot of room for future potential investigative efforts. And network studies themselves have demonstrated that asthma microRNAs may serve as key hubs regulating both genes and network pathways. So let's talk a little bit about tissue-specific microRNA functions. We just mentioned the Th2 cell, which is over here in the context of allergic rhinitis. And there actually have been microRNA studies done on almost every asthma cell that certainly is on this list. I wanted to just, for example purposes, focus on two cells in particular, a study that was done in airway smooth muscle cells and one that was done in pulmonary macrophages as they relate to asthma. So this study was performed via sequencing of small RNAs in airway smooth muscle cells. And through those sequencing efforts, they noted that microRNA 10A was the greatest, the microRNA with the greatest expression in these airway smooth muscle cells. So much more prevalent than any of the other microRNAs. And indeed, the miR10A accounted for approximately 25% of all the microRNAs that were noted in this study. Now, when they looked at other microRNAs in terms of its expression across other cells, they noted that microRNA 10A is actually very specific to airway smooth muscle cells. There was some expression as well in airway fibroblasts, but if you look across the board at all these other cells, microRNA 10A expression really seemed to be pretty doggone specific to airway smooth muscle cells. And this contrasts tightly with another microRNA that was expressed in airway smooth muscle cells, LET7A, which is pretty universally expressed across a wide variety of tissues. Now, in order to investigate the potential for function of these microRNAs in airway smooth muscle cells, these investigators, Chun Liu and his colleagues at the School of Public Health here, really looked at cellular proliferation as augmented and DNA synthesis as augmented or inhibited by microRNA. And so they administered microRNA mimics to these growing airway smooth muscle cells, and they noted that the miR10A mimics actually decreased cellular synthesis across time. And the decreased cellular synthesis, it looks like, via inhibition of DNA synthesis, in particular by specific microRNA 10A inhibition, decreasing DNA synthesis. And if you gave an antagonist or an inhibitor of microRNA, you increased DNA synthesis in this airway smooth muscle cells. By using informatics, you could look at the targets of microRNA 10A. And one of the targets that came up was PIK3CA. PIK3CA is specifically known to inhibit cellular function in terms of overall replication. And so a hypothesis was performed that perhaps microRNA 10A was functioning through inhibition of PIK3CA. And indeed, when you administer mimics and inhibitors of microRNA 10A, you show great changes in PIK3CA protein expression up and down, suggesting that miR10A may, in fact, regulate airway smooth muscle function through a PIK3CA mechanism. Okay? Let's also talk a little bit about miR9 in pulmonary macrophages. And the reason that I wanted to talk about this is that this is an example of a microRNA in a very context-specific fashion. In this case, the authors were very interested in the fact that in cellular models, if you combine interferon gamma and LPS, you get a model of corticosteroid-resistant asthma. Now, interestingly enough, if you look at LPS and interferon gamma, either by themselves or under vehicle control levels, miR9 is not very well expressed at all. But if you give this inflammatory condition, microRNA 9 goes way, way up in terms of its overall expression. Interestingly enough, if you look at asthmatic sputum from neutrophilic predominant asthmatics, which tend to also be corticosteroid resistant, miR9A is also much higher expressed than in eosinophilic predominant asthma. So the hypothesis really was, well, maybe microRNA 9 is a context-specific inhibitor of glucocorticoid resistance. Okay? And the investigators actually went about to prove that by administering antagonists, or anti-microRNA 9s, and they looked at it in the context of mirroring models of airways resistance. This is an ovalbumin model. And you can see that particularly in the case of administering antagomir of miR9 antagomir, nothing happened in terms of airways resistance. However, if you added glucocorticoids, dexamethasone, and you administered miR9A antagomir, so dexamethasone itself still had a lot of airways resistance. If you add the antagomir along with dexamethasone, you actually restored airways resistance back to normal. So context specific in terms of microRNA 9 works via inflammatory conditions to restore glucocorticoid sensitivity. Now, these authors further said, well, what is the mechanism for this? And again, they looked at an informatics approach, and they were able to map microRNA 9A as regulating potentially PPA2 activity. And these are PPA2, the two subunits of PPA2. PPA2 specifically has been implicated in regulating glucocorticoid receptor nuclear translocation. And when they looked at both of the subunits, actually a miR9A inhibition actually miR9A mimics, resulted in decreased expression of both of these subunits of PPA2, suggesting that miR9A may in fact be regulating PP2A activity by its subunits. The authors further said, well, we know how to look at nuclear translocation of the glucocorticoid receptor. We can do these immunofluorescence studies as well as these other studies. And in fact, if you look at just dexamethasone alone without the interferon gamma model, yeah, of course you get nice expression of glucocorticoid receptor within the nucleus of these cells. If you administer the inflammatory model, you see that glucocorticoids are really not incorporated whatsoever, even after the administration of dexamethasone. But if you give the antagomir, you restore glucocorticoid incorporation despite having this inflammatory model of interferon gamma and LPS. They further said, well, what if we just give PPA2 directly to further enhance the proof of the mechanism? And indeed, PPA2 administration directly in the context of this inflammatory model also restored glucocorticoid sensitivity overall, therefore leading to an enhanced overall model saying that miR9A clearly regulates PPA2 via one of its subunits to augment glucocorticoid sensitivity. There have been a lot of other microRNA cell-specific studies. This is just kind of a broad cartoon overview of specific microRNAs that have been proven to regulate specific genes in the context of asthma. So, in summary, microRNAs in asthmatic cells, certainly individual microRNAs can be ubiquitously expressed, such as LET7A, but they can also be very tissue or condition specific. We talked about one tissue-specific condition with miR10A being very specific for airway smooth muscles, and we talked about a condition-specific expression of miR9 as it relates to interferon gamma LPS stimulation as a glucocorticoid resistance models. Now, these microRNAs can target specific genes to clearly influence asthma cellular phenotypes and treatment responses. Let's talk a little bit about the area of microRNAs that I'm most interested in is circulating microRNAs. Now, how are these things circulated? Well, we mentioned before that, active cells secrete microRNAs via exocytosis and small vesicle production, whereas necrotic cells produce microRNAs by releasing them freely where they're bound to serum proteins. And then they're freely circulated either within these bodies or they're circulated bound to the arginate and HDL proteins. We mentioned before that they are crucial regulators of both local and long-distance cellular signaling, and I just wanted to reiterate that is why these may be such important biomarkers, is that they can be measured as they're in their intermediate phase of cellular signaling. Now, the cool thing about microRNAs in the serum is that they are packaged. We talked about nanovesicles, we talked about complexing with protein complexes, et cetera, but because of the fact that they're packaged, they are not subject to RNases, which normally breaks down circulating RNA. And the reason we can't often measure RNA in blood is because they're rapidly degraded. As evidence of this, these investigators actually looked at stability of microRNA in as many as 5 years after storage at minus 20 degrees Celsius. And you can see that, for the most part, across the board, whether it was exosomal microRNAs or plasma-bound microRNAs, the expression of microRNAs was very stable over time. The one exception here was if you don't store the sample well, if you just store it at 4 degrees Celsius, room air for 2 weeks, then you can get some microRNA degradation. But as long as the samples are stored well, the expression persists over many, many years. Now, this study was published last year. Brigham and colleagues really said, well, can we use circulating microRNAs to help differentiate healthy individuals versus allergic rhinitis versus those with asthma? The healthy individuals are shown in gray, allergic rhinitis in blue, and the asthmatics in red here. And again, there's differential expression both from healthy versus both disease subtypes, but as well as condition-specific differential expression of these circulating microRNAs. The authors then said, well, if we look at the specific conditions, what are the pathways and particular genes that are regulated? And each of these groups really refers to, in this case, the microRNA was more heavily expressed in asthma versus allergic rhinitis, but both were expressed differentially versus healthy controls. This is asthma alone. Group number 3 was allergic rhinitis alone, et cetera, et cetera. Okay, so differential conditions, and you have differential downstream pathway genes that are specifically inhibited in relation to this microRNA expression in the circulation. They also said, well, what if we look at the asthmatics themselves? Can we get a better idea just by looking at the circulation what the microRNAs are doing? And indeed, they showed the 2 groups of asthmatics that were strongly differentially expressed by microRNAs, and in fact, these groups defined eosinophilic versus non-eosinophilic asthma, suggesting that even within asthma itself, microRNAs may be useful in helping to do genomic subtyping of groups. The final thing they did is said, well, we have all this microRNA data. Can we use this for prognostic classification? Can we accurately identify who was in what group just by using microRNAs? They used 6 microRNAs, and they used a random forest algorithm to try and predict which person was within which group. And they accurately classified 72 of the 79 participants that they had. And when they looked at it from an AUROC, the area in the ROC curve classification perspective, they had a 97% accuracy for the classification using these microRNAs. Of course, this is cross-sectional data, but it actually is very good to be able to distinguish healthy versus disease groups just based on that, suggesting that perhaps these can be used as clinical biomarkers. Now, our efforts into this really look more at asthma severity rather than asthma case control status. And for this, we've been interrogating microRNAs in the Childhood Asthma Management Program. The CAMP was a 4-year clinical trial study that randomized budesonide versus placebo versus nidocromil therapy. And the helpful thing for us is not only were these individuals followed very carefully for 4 years, but they were also followed for 16 years as part of an asthma clinical cohort study subsequent to that. So we followed all of these children until they were adults as well. Okay? MicroRNAs themselves were obtained right here at the time of randomization, allowing us to look longitudinally to say, are there baseline microRNAs that might predict longitudinal characteristics? So we did a serum microRNA study. At this point in time, it was 160 camp serums. They were all allocated to the budesonide group and all from Caucasians. Our initial studies have focused on this TACMAN microRNA panel, which has 754 microRNAs. More importantly, of those 754, about 15% were well expressed in the serums of these asthmatics, and at least 75%. And interestingly, of those 108 microRNAs that were expressed in these known asthmatics, 73 or 67% had been associated in at least one prior study of asthma susceptibility, suggesting that asthmatics don't just circulate microRNAs from everything, they tend to circulate microRNAs that are asthma microRNAs. So we looked at this in the context of just a differential expression asthma versus no asthma study. We used a publicly available data set that had the same type of sample, serum, and used the same platform. And we were able to say, yeah, based on our asthmatic microRNAs, we had 76 of the 108 microRNAs that were actually differentially expressed compared to these healthy controls. Again, suggesting that the microRNAs that we were expressed in our asthmatics were asthma-specific, 76 of the 108. If you look at this from a cross-sectional perspective, we have perfect prediction and classification as to who was asthmatic versus not asthmatic just by using 3 microRNAs. But more important, perhaps, is how does microRNAs function to perhaps influence asthma severity and longitudinal phenotypes? I'm just going to give you a broad overview. We looked at this, and we've published on microRNA and lung function. Of the 108 circulating microRNAs, 22 of those microRNAs were associated with the FEV1 to FVC ratio. 4 were associated with just FEV1 by itself, and 8 were associated with FVC. This is just an example from our paper. We showed that miR27B, which is a previously associated asthma microRNA, is strongly correlated with FEV1 as a percent predicted. We could scale the axes, but obviously this is a 20% FEV1 decline, which is relatively significant in terms of the overall perspective. miR15B, on the other hand, is another microRNA that's been previously associated with asthma. And again, you can see that this is associated with the FEV1 to FVC ratio, suggesting that microRNAs can help to modulate asthma severity. In terms of pathway analysis, these are the pathways from the predicted gene targets from the microRNAs that we've had. And you can see that they correlate to relatively well-known asthma pathways, including G protein-coupled signaling, SMAD proteins, and PPAR gamma signaling, as well as some, since this was a lung function study, as well as actin and myosin, so smooth muscle function as it might relate to airway function. What we're most interested in, though, is can we prognosticate? Can we use baseline microRNAs to say what is going to happen with our asthmatics in the future? So we know that as part of an entry criteria for camp, everybody had significant airways responsiveness with a PC20 less than 12.5. I mentioned that these subjects were followed longitudinally for 16 years, and during that 16-year time frame, 32 of our 160 asthmatics with microRNA data underwent airways hyperresponsiveness remission, where their AHR was now not detectable. Okay? So we did what's called a Bayesian network to try and classify, can we predict who these asthmatics were that underwent remission? And this looks like it's fairly complex, but basically the bottom line is that we were able to generate a group of 12 microRNAs that specifically predicted who was going to undergo longitudinal remission in their airways responsiveness as late as 16 years after the sample was taken. When you look at it in the more traditional AUROC curve, This curve, the top curve, is actually incorporating baseline PC20 because actually if your PC20 was on the higher side, you were in fact more likely to have airways responsiveness remission. But if you incorporate that both along with the PC20, we had an AUROC of approximately 87% to predict who is going to have remission of their airways responsiveness versus not. And this paper is currently in submission. The other thing, obviously, we're all interested in asthma exacerbations here. And so we also looked at 1-year exacerbations defined as who in the CAMP program had significant exacerbations requiring at least a prednisone burst to be prescribed. And 38 individuals underwent a significant exacerbation within the first year after randomization. The one thing that we wanted to do is say, well, can microRNAs actually do better or improve on a published clinical asthma exacerbation score? This actual exacerbation score was actually published in relation to the CAMP population. Part of the reason that this does not reach significance is you'll recall that all of the microRNA populations that we had were from the inhaled corticosteroid group. So there was some attenuation in terms of overall exacerbations, but this overall model was tested in a broad CAMP population and was significant there. So, are microRNAs associated with longitudinal exacerbations? Well, in fact, we found a dozen microRNAs that were strongly associated with asthma exacerbations over time, and there was anywhere between a 20 and 40% risk reduction of exacerbations despite being on inhaled corticosteroids added to this by having the microRNAs. When we look at this from a prognostic perspective, can microRNAs improve utility over clinical models alone? Well, we see that in this particular case, the AUROC for the clinical model alone was 0.67, so it's only had modest predictive benefit. MIRS did a little bit better, but not a whole lot better. But actually, if you combine a clinical score with microRNA testing, you actually get to the point where you have good prediction for who is going to have exacerbations within the year after after randomization in this particular clinical trial. So, what can we say about circulating microRNAs overall? Well, they're stable. And that's great because you can study them. In this case, the CAMP samples were taken 20 years ago, and we're looking at microRNA expression in these individuals. They do reflect the tissue of origin, and we showed that basically all of the microRNAs were either in our CAMP samples, were either differentially expressed or they had been previously associated with other asthma studies. And there is increasing evidence that microRNAs can serve, to me, as very useful biomarkers for asthma disease susceptibility, severity, and the natural history. So the last topic that I want to talk about is microRNAs as potential therapeutics. Now, unfortunately, the cardiovascular folks are a little bit ahead in this realm, so this is a slide taken from Circulation Research, not typically a journal published here at Asthma Grand Rounds, but just to say that this company is actually working on therapeutics using microRNAs to address all of these areas in cardiovascular pathology. Some areas, certainly including inflammation and fibrosis, as well as metabolism, smooth muscle function, things along those lines, are all potentially very relevant to asthma as well. So what can we say in terms of what have we done so far in terms of therapy for asthma? Well, everything that's been done has been done so far in mouse models, and I'll say that right up front. So I'm going to present two examples of mouse models treating mice with microRNAs. This is a house dust mite-induced model of microRNA 126, and you can see that in this particular house dust mite, they found 3 microRNAs that were strongly differentially expressed between the house dust mite-induced mouse model versus the regular mouse. And in fact, when they looked at that in the context of knockout mice, TLR4 and MyD88 both decreased the expression of miR126. So they were actually very interested in this as a context-dependent microRNA. They looked at this, then they gave an anti-MIR, and they administered the anti-MIR 126 via the nasal passages of the mice, and they looked at a wide variety of outcomes, including airways responsiveness, including inflammatory cell production, as shown both up here and down here. And they found, indeed, that giving anti-microRNA 126 intranasally in these mice did result in significant attenuation of airways responsiveness and did, in fact, result in significant decreases in inflammatory cell exudates. One thing that they also noted is that they might have a specific mechanism for this MIR 126 effect, because when they looked at this specifically in house dust mice stimulated individuals, Th2 signaling cytokines were markedly decreased, as seen here, and the amount of CD4 positive cells were actually also decreased. In addition to the MIR-126 model in house dust mite, other folks have looked at ovalbumin models of murine asthma. This is an ovalbumin model just basically saying, well, if you look at sequencing data from the lungs of these mice, you can see that naive mice of the lungs don't actually have all that many microRNAs, and microRNA production goes way up as soon as you give these mice ovalbumin-induced airways responsiveness. Okay? And of all these microRNAs, the most abundant one that they found was LET7. Now, LET7, indeed, from its target seed sequence, is predicted to directly inhibit IL-13 production. And in fact, when they looked at this in terms of administration of LET7A, it was specifically anti-correlated with IL-13 production, suggesting that higher levels of LET7A are correlated with lower levels of IL-13 and higher levels of interferon gamma, and vice versa. So what they did specifically after that was they formally tested this hypothesis about LET7A and IL-13 production by giving MIR mimics and antagonists, and they found exactly the same thing, that IL-13 production did in fact increase in the, when given antagonists, and IL-13 production decreased when given MIR mimics in these HEC293 cells, both derived from murine bloodlines as well as human bloodlines. While those particular authors did not test LET7 administration, another set of authors actually did look at this in the context of administration in this murine ovalbumin model. And again, we predicted LET7 should decrease IL-13 and therefore help to decrease this inflammatory process. And in fact, that's what we saw in terms of the LET7. Inflammatory exudates decrease, airways responsiveness decreases as well, and overall inflammation score goes way down with the intranasally administered LET7. Okay? So we just talked about LET7. These are current MIR therapeutic companies in it over and above the anti-MIR therapeutics. There are 3 companies that are actively pursuing things. LET7A is actually in the active development pipeline. I don't believe it's in the active development pipeline for asthma, but at least there, at some point in time, will likely be preclinical trials going forward to say, is LET7A potentially viable for use in therapy? So, what have I talked about today? Well, in terms of microRNA and its relation to asthma, we know that microRNA is broadly associated in airway cells, inflammatory cells, and within the circulation itself. We talked about the fact that microRNAs, because one microRNA may influence the gene expression of many genes, can serve as key network hubs for broad regulation of asthma-related pathways. And this may give us some insights going forward into broad biologic understanding that we haven't had to date specifically related to these microRNAs. We also have noted that there are some times when microRNAs can be very tissue-specific or condition-specific regulators, and these can therefore help to facilitate specific intracellular processes. Now, circulatory microRNAs, to me, show broad promise as prognostic biomarkers. True longitudinal studies, this is what I'm funded to do, we are doing deep microRNA sequencing in literally hundreds of asthmatic samples to really look at the prognostic implications for what can we do with these microRNA samples. We also know that microRNA mimics and antagonists are being actively developed by companies for clinical therapy for non-asthmatic diseases. And we have shown, at least in murine models, that microRNAs can be of benefit if administered intranasally as a therapeutic adjunct. So there certainly is potential, at least, for clinical translation for asthma for the future. And you may envision something like this in the domain of saying, okay, well, in the future, might it be that a patient comes in to see a physician for asthma, they obtain blood. In this case, you can also measure microRNAs in urine and other body fluids as well. From that, do microRNA profiling, help with the diagnosis as well as prognosis overall of lung disease, and then could that lead to MIR-specific inhibition therapy? So I'd like to acknowledge all the folks that are part of our microRNA working group at the Channing. These folks are listed over here in my funding through the NIH, and I'm happy to take any questions in relation to this talk. Thanks.
Thank you very much. It's a window into a brave new world, it feels like to me. Questions?
Yes. Do you have any insight about the role of microRNA for no-allergic asthma?
For non-allergic asthma? I think that's a really good question, and you know, most studies have not specifically differentiated allergic asthma versus non-allergic asthma in the literature yet. We showed the one study by Panagraban and colleagues that was the circulating microRNAs that clearly show that there is differential expression of microRNAs for neutrophilic predominant asthma versus eosinophilic predominant asthma, and I think that gives us some insights there. We've also begun to say, well, you know, are there microRNAs that are specifically associated with other, you know, atopic asthma phenotypes, you know, so you can look at, for instance, eosinophilic predominant asthma or asthma where you know people have allergic predominance and things like that. But I think all of those studies are in their infancy, to be honest with you, and there's nothing that I've seen specifically in the literature outside of that study that I showed that says differential microRNA expression between neutrophilic and eosinophilic predominant asthma. Yeah.
This is so interesting. What's the thought about as control or exacerbations occur, whether levels of microRNA change? I know you talked about the stability of the microRNAs overall, but is it thought that they're stable throughout time and control levels and exacerbations, or is there any data?
No, I think that's a very good and very intriguing question. I think that nobody, I will say again, nobody has specifically looked at that. We know by regular gene expression studies that there are many, many, many genes that are differentially expressed during acute exacerbations of asthma. And so as an extension of that, it would be natural to think that there will be some differential expression during the acute process, and understanding that differential expression of microRNAs during acute exacerbations may also give you differential insights into potential for therapy versus not therapy, as compared to looking at just baseline, what are the predictors along those lines. So I think that it's a fascinating question going forward. Unfortunately, there are very few studies that actually get the appropriate sample types for you to be able to specifically analyze that. There are some publicly available data sets where they have actually done yeoman's work in terms of looking at gene expression data, and I think that using informatics processes, you could at least back infer what are the likely microRNAs that are upregulated specifically during those inflammatory exacerbation phases versus not, and it's a great idea in terms of something to do.
You know, when we do translational, study, what would you suggest, do you suggest it will be an intradermal injection, IV injection, or what kind of formulation you anticipate?
I love this question. I think that most people who have actually looked at this from a pulmonary perspective are very interested in those exosomes, those small nanoparticles, which are basically, because we know that they can be endocytosed. And there have been a lot of studies that have been really focused in the pulmonary literature as well as other literature, trying to focus on how can you get nanoparticles to be tissue and target specific in terms of their uptake. And they've made a lot of progress actually with that. They haven't incorporated specific microRNAs or anything like that within the nanoparticles. And I think that part of things probably wouldn't be all that hard. But there is a whole huge focus on, can we generate these small particles that can be actively uptake within the airway? And the few studies that have looked at this in the context of the lung have actually done that via nebulization, not surprisingly. And so I think that at least the earliest phase studies that you'll see that are lung-specific or asthma-specific in this case will likely be nebulized studies as they're performed on nanovesicles.
But if you compare to previous failure, for instance, like inhaled insulin.
Yeah.
So I would say we learned something, from our other department when we try to translate, use the same delivery system.
Right.
So there may become some kind of, surprise side effect when they pop up.
Absolutely. I think the concerns are widespread in terms of trying to know what side effects there are. So I think addressing two things. One is insulin's a little bit different because you actually want that to get into the bloodstream. And in this case, you'd want whatever you're administering to not get into the bloodstream and to be locally derived and to stay local. I would say that there are also concerns about microRNAs because they are so pervasive. I mean, if you're regulating dozens to hundreds of genes with a single microRNA, the potential for off-target effects is clearly there. And so the thought is do you use microRNAs themselves and anti-microRNAs themselves, or do you do something which is more synthetically derived where you actually have more of the siRNA type of phenomena, short RNAs that are much more specific for specific genes? Sure.
Is there any data on how respiratory pathogens may influence microRNA?
There are data in terms of the microbiome in general in microRNAs. And I think that there are two interesting contexts for microbiomes in terms of small RNAs in general. One is so your specific question about respiratory pathogens influencing microRNAs, very few of those studies have been done. There have been a couple of studies looking at Pseudomonas aeruginosa, so more from a pneumonia infectious perspective and differential microRNAs, but certainly in the asthma side of things from the asthma microbiome, that has not been done. In the broader context of the gut microbiome, folks have done some associative type of studies, looking at clades within the gut as they relate to differential microRNA expression. So those types of studies have begun. I think the one really interesting context from a broader study perspective really is, and something we've been interested in, is if you measure and sequence small RNAs, including microRNAs, you can actually measure and sequence the circulating microbiome as well. And so I think that one of our near future goals is to try and identify what actually is circulating and how does that correlate with microRNA expression. Josh.
So following up to Katie's question,
Yeah.
Are there microbially derived miRs that alter human gene expression, and are those relevant to asthma?
I think that that's a great question, and I think that we're early enough into this that I don't know that anybody has looked at that certainly not within an asthma context, but within any disease context. Clearly there are small. We said that the microRNAs are conserved across species, and there are clearly bacterial-specific specific microRNAs that may not be that specific because of the conservation. In theory, lysis of a bacteria might produce microRNAs that may readily actually bind and regulate human disease and cause differences in pathogenesis. So I'm actually not sure. I think it's a broad, open area.
Great. Food for thought and research. Thank you very much. Terrific. Nice stuff.